So, ladies and gentlemen, we will start shortly. Good day to everybody. Good morning, people in the Western time zone, Pacific time. Welcome to people in the Eastern time zone. We're delighted to welcome Spectradyne and colleagues for the webinar today. Today's webinar was going to be led by Dr. Jean-Luc Fréquin, the CEO of Spectradyne, who will give an introduction. Following that, ladies and gentlemen, we're going to have a number of guest speakers, excellent academic guest speakers that Spectradyne has invited who are going to speak. After that, Dr. Fréquin will give a wrap-up of the webinar. And then subsequently, we'll have a live Q&A as we've done in past years. So without any further ado, ladies and gentlemen, right on schedule, we'd like to welcome Dr. Jean-Luc Fréquin, CEO of Spectradyne, to get started and to give his opening statement, introduction to the technology before we get started with the guest speakers. So welcome, Dr. Fréquin.
Great, thanks, Anil. Welcome to everyone attending, and thank you all for joining us for today's webinar. We have an exciting lineup. We'll learn directly from two experts about how extracellular vesicles can carry spike protein from COVID-infected cells and the roles that EVs play in vector-borne disease transmission. I think we'll learn some cool things today that hopefully can be applied to your own research, but at the very least, it'll be something interesting that you didn't know before. I'll start by giving a short introduction to Spectradyne's technology, which is called Microfluidic Resistive Pulse Sensing, or MRPS, and which features in some of the experiments we'll see today. Then I'll hand it over to our guests. First, Dr. Zach Troyer from the Whitwer Lab at Johns Hopkins will present a paper from his PhD work entitled EVs Carry SARS-CoV-2 Spike Protein and Serve as Decoys for Neutralizing Antibodies. Dr. Troyer received his PhD from the lab of John Tilton at Case Western recently. Second, we'll hear from Dr. Hameeda Sultana, who recently moved from Old Dominion University in Virginia and is now associate professor at the University of Tennessee Knoxville. She'll present a selection of results about exosomes and vector-borne diseases, and I know it's going to be a fascinating presentation. Now, common to each of these presentations is the importance of being able to measure the concentration of extracellular vesicles accurately. And this typically comes up in three areas of EV research. First, when EVs are being used as therapeutics, so nanomedicine applications, being able to measure the concentration accurately leads to faster development and ultimately safer and more effective products. Second case we commonly see is when EVs are being used as an assay readout, so as a biomarker. And in this case, the concentration really is the measurement, the outcome of the experiment. And so the better you can measure concentration, the greater sensitivity and specificity that you end up with in your assay. Finally, in fundamental EV biology, and this is where we'll hear about today, it's really important to be able to measure EV concentration accurately, at the very least for normalizing for quantitative experiments. So for example, if you're studying maybe EV regulation in a biological system and you stimulate two models and you want to quantitatively compare the EV output from both of those different models, you need to be able to normalize by the concentration of EVs that you're measuring first. So if you're doing proteomics, for example, you have to put in the same amount of starting material, right? So ultimately, being able to measure the concentration of EVs more accurately leads to clearer experimental outcomes, and that's faster and easier science, which is what we all want. So there's a critical need, clearly, in EV research for accurate EV concentration and size, and that will be a common theme in all the presentations today. That's also where Spectradyne comes in. This is where our technology shines. And so I'm going to spend a minute reviewing it because it features in the papers today. And so we're all on the same page. What I'm showing you here is Spectradyne's NCS1 instrument. It's a small benchtop instrument that is cutting edge technology, measures EV particle size and concentration, starting at about 50 nanometers diameter up to about 10 microns in diameter. And really importantly, it does not use optical techniques to measure the particles. So it uses electrical detection. It's based on the historically tried and true Coulter principle. And importantly, if you're following MySEF guidelines, this qualifies as a, the term they use is complementary technique, right? Because it offers a fundamentally different perspective on the particle samples than the optical techniques like NTA, for example. Plus, it gives more accurate concentration, but that's a different topic. It's fast and easy to use, and it's easy to adopt. And it uses microfluidics, which gives a key advantage that we'll hear about today, I think, from Dr. Sultana. Only 3 microliters of the sample are required for analysis. So that enables crazy science like measuring EVs in tick saliva, for example, which I hope we'll get to hear about a little bit. Let me spend 1 minute describing how the NCS-1 works, and then we'll get to the speakers. So the NCS-1 uses Microfluidic Resistive Pulse Sensing, or MRPS, to count and size individual EVs in a sample. And all the magic happens in this microfluidic cartridge I'm showing you here. It's about a ½ inch on a side. You pipette your sample into the reservoir, and then we flow it through the cartridge where particles flow one by one through a bottleneck in the fluid flow, highlighted in yellow here. And we're making electrical measurements, so every time a single particle passes through that constriction, we get an electrical signal. And from that, we measure we measure 2 main properties. One is how big the particle is. That's given by the amplitude of the electrical signal here. And we also measure how fast the particle goes through the constriction, because that tells us how much sample we're measuring of the volume flow rate of the sample. And that allows us to deliver absolute concentration measurements. So if we measure for longer, this is real data now, 60-second measurement showing detection of many particles. You can see just straight in the raw data here that the particles take on 4 different sizes in this sample, as shown by the clustering of the red dots that are the particle events. You can see that in the histogram of just counts, particle counts, but we can go a lot farther than just counting particles. We can measure the absolute concentration of them because we know the volume of the sample that was measured in which these particles were counted, right? So we can convert counts to concentration. And this is what our technology typically delivers. So this plot is showing concentration on the y-axis versus particle diameter on the x-axis. And this sample in particular was a mixture of polystyrene particles, 50, 90, 120, and 150 nanometers in diameter. And this technology is capable of quantitative concentration measurements over any sub-range here that you want. You can just click and drag essentially to measure the concentration over a given size range of interest. So this combination of accurate concentration and size together with the ease of use of the platform is really not available with any other method right now. We invite you to come to our website to learn more. We have a lot of resources available about particle analysis generally and specifically about different applications. And if you're really curious, we invite you to send us a sample. We're happy to measure 1 or 2 for free to demonstrate the power of our technology. So please reach out to us. With that, I will turn it over to Dr. Zach Troyer from Johns Hopkins, who will present extracellular vesicles carry SARS-CoV-2 spike protein and serve as decoys for neutralizing antibodies. Thanks, and thanks for joining us, Zach.
Thanks, Jean-Luc. Just need to share my screen real quickly. So I'd like to thank the organizers of the webinar today for giving me the chance to speak about my work. And I'd also like to say that any opinions that I express in this talk are not necessarily representative of those of my current lab or institution. So the work I want to present today is work that I did during my PhD in the lab of Dr. Chip Tilton at Case Western Reserve University. The title of our article that we had published in Journal of Extracellular Vesicles a few months ago is called Extracellular Vesicles Carry SARS-CoV-2 Spike Protein and Serve as Decoys for Neutralizing Antibodies. So just to give a little bit of background about the research interest, the things I'm interested in, I'm really interested in extracellular vesicles and viruses and the intersection between extracellular vesicles and viruses and how they might interact. But to give a little bit of background about extracellular vesicles, we always want to know what are extracellular vesicles. How do we define them? Well, they're diverse lipid bilayer enclosed nanoparticles that are released constitutively by most cell types. There are two major release pathways that have led to historically two major classifications of EVs, exosomes and microvesicles. However, more recently, rigorous characterization has revealed that there are many subtypes of EVs outside of that initial classification. But across all of that, there's always been some major protein markers that have been considered EV markers, and these consist of the tetraspanins CD9, CD63, and CD81. These are transmembrane proteins. So as I said, I'm also interested in viruses, so I wanted to talk a little bit about viruses and their structure. Typically, viruses can be enveloped or non-enveloped, whether or not they have a lipid bilayer derived from a host cell. Now, enveloped viruses in some ways are very similar to EVs. And I'm giving a picture of a very physiologically relevant envelope virus here that many of you probably have heard about, SARS-CoV-2, which has caused our recent pandemic. And typically a viral structure consists of various viral proteins. If it's an envelope virus, it will have that lipid bilayer I mentioned. But one very important protein I want to point out is something called the viral fusion protein, which typically is a transmembrane protein that sticks out of the viral envelope. And for the case of SARS-CoV-2, this is the spike protein. And this spike protein has two subunits, S1 and S2. Now, these viral fusion proteins for most viruses are responsible for the viral attachment to the cell it's trying to infect, and then for the membrane fusion that occurs after that attachment, allowing the virus to deliver its genetic material into the infected cell. So these proteins are, as a result, very important for the viral life cycle. And similarly, because of their importance, many of the host neutralizing antibody responses are directed towards these viral fusion proteins. The SARS-CoV-2 vaccine, or the COVID-19 vaccine, generates neutralizing antibodies against this spike protein as a result. Now, viruses have some defenses against these neutralizing antibodies. For example, HIV produces a large amount of immature, non-properly processed HIV particles that are non-infectious, but they can strongly bind up host neutralizing antibodies, allowing the actual infectious virions to escape detection and neutralization. HPV does a similar thing with what are called subviral particles, which are just smaller sized viral particles that are non-infectious again, and they carry the viral fusion protein of HPV. So similarly, it's a defense against neutralizing antibody responses. And this is just kind of a graphical representation of what I'm talking about. Basically how viruses can circumvent the adaptive immune system and neutralizing antibodies. You have a infectious viral particle here, a neutralizing antibody from the host here, and some sort of subviral particle or defective particle that blocks the ability of the neutralizing antibody to act on the infectious particles by binding it. And this leads to effects of increased infection and decreased neutralization. So I'm talking about EVs and viruses here, and it's, as I mentioned before, EVs and viruses actually have substantial overlap, especially envelope viruses. And while they are distinct particles, there is a distinct virus and there is a distinct host EV that doesn't contain any viral proteins. There's also a gradient of particles that exist between coming out of an infected cell that, like, for example, a virus-induced EV that can carry various viral proteins. This isn't a virus, it's an EV, but it has a viral protein or perhaps a viral RNA inside of it. And so these are the types of EVs that I'm very interested in, these virus-induced EVs that are carrying viral proteins. And interestingly, there are many instances in which these virus-induced EVs are carrying the viral fusion proteins. And I just have many examples from literature here where EVs have been shown to carry these viral fusion proteins from various types of viruses, such as vesicular stomatitis virus, influenza, HIV, and the human CMV virus. So our question coming into the pandemic, it was an opportunity for us, was to ask, what about the SARS-CoV-2 spike protein? Is it possible that this viral fusion protein ends up on the surface of EVs, and what effect might that have? And just to give an example of why we think this is interesting, we thought of a scenario that could potentially occur if EVs are able to carry the spike protein. Normally with just the SARS-CoV-2 virus present, you'll get infection of the airway epithelial cells, or whatever cells the virus is infecting. In the presence of an anti-spike neutralizing antibody, you should get neutralization and there will be less infection or no infection of these cells. However, potentially, in the presence of EVs carrying the spike protein, if this is something that occurs, they could potentially bind the neutralizing antibodies and reduce their efficacy at blocking the actual virus from infecting cells. So this is something we would call, if we find it, the EV decoy effect. And so we were looking for this effect to see if it occurs and to see if the EVs carry the spike protein in the first place. So to look at this question, we needed to produce extracellular vesicles from cells that are expressing the spike protein. So we had some HEC293 T cells that we grew in EV depleted media, and we either mock transfected these cells or transfected them with a codon optimized version of the SARS-CoV-2 spike protein. We then isolated the conditioned media from those cells. We filtered it to remove cell debris, and then we processed it with tangential flow filtration to purify and concentrate those EVs. So that gives us a nice population of purified EVs, which we can then use in our various characterization methods, which one of those, of course, is the microfluidic resistive pulse sensing that Jean-Luc has talked about, alongside other methods such as nanoscale flow cytometry, electron microscopy, and various proteomics. Following our characterization, of course, we looked at the functional effects of these EVs in reducing spike-mediated viral infection. So our first main figure here, we were looking to see if the EVs carry the SARS-CoV-2 spike protein to begin with. So for this, we used a EGFP tagged version of the spike protein, and we expressed it in these HEC293 T cells, and we saw a nice ergic localization of the EGFP signal, which is consistent with the known localization of spike protein. And we also, I should say, transfected our cells with fluorescently tagged versions of these EV marker tetraspanin CD9 and CD81. So we were looking to see if our EVs would have co-localization of these fluorescent signals, which was just a EV carrying a EV marker and the spike EGFP. So when we did these nanoscale flow cytometry experiments, we did see a nice double positive population of CD9 positive or CD81 positive EVs that were carrying spike protein. So this shows us that the EVs do have the capacity to carry the spike protein on their surface. Now, of course, that was an EGFP tagged version, so we moved then to using a non-tagged version, a wild type version of the spike protein. And we produced those EVs as before, and then we characterized them again. This time with electron microscopy, we can see our EVs here. And then we used the NCS1 instrument and the MRPS technology to get concentration and size data of our extracellular vesicle preparations, and of course, to normalize experiments downstream. And we also did the same with some lentivirus that I'll talk about in a bit. We also did proteomics characterization to find the wild type spike protein, whether it's present on the EVs. And with the mass spectrometry analysis, we did see that the spike protein was present on the EVs. And when we did Western blotting, we actually saw something quite interesting, which was that the majority of the spike protein we detected on the EVs was actually the S2 subunit, which is the transmembrane subunit, and it was missing the S1 subunit. However, we did see a faint band for the full-length spike protein. So moving forward, we wanted to test the effects of these spike EVs in their ability to influence infection and neutralization. So for this, we needed to have some method of looking at spike-mediated viral infection. Now, we used a SARS-CoV-2 spike HIV pseudotype, or an SLV, basically consisting of an HIV core and then the spike protein on the envelope. So the infection itself would be mediated by the initial steps of infection would be mediated by the spike protein. And within our HIV core, we have this beta-lactamase protein, which is important for our assay. So we load this dye called CCF2AM into our target cells. This dye consists of two fluorescent moieties that are linked by a beta-lactam ring, and they form a fret pair. So when we excite it with a violet laser, we get a green signal coming out of the cells. However, if this dye gets exposed to beta-lactamase, the fret pair is cleaved, and now we get a blue signal under the same laser excitation conditions. So if our virus is able to infect the cell and deliver its contents, including beta-lactamase, into the cytosol, we will see this blue signal. We'll see the cells convert from green to blue. So that's how we measure our spike-mediated infection. Now, we needed to test our system of infection to make sure it's actually working the way we expect. And so we used target cells, which were HEC293 T cells expressing the ACE2 protein, which is known to be the receptor for spike. And you can see here that we get an infection signal or a fusion signal whenever we add our spike lentivirus to the ACE2 expressing 293Ts, and this does not occur if the cells are not expressing ACE2. We then obtained a commercial neutralizing antibody against the spike protein, and we added it into the system to see if we could knock down that level of infection. And indeed, what we found is that we were able to reduce the infection and show that this antibody can neutralize the viral entry and the infection. And we similarly showed this neutralization effect at a range of concentrations here. Now that we have a working model of spike-mediated viral infection, we wanted to look at the influence of our spike-carrying EVs in the system. So our hypothesis was that with increasing amounts of these spike-carrying EVs, we would see reduced levels of spike lentiviral neutralization and an increase in infection. Basically, the antibodies would get would be worse at doing their job in the presence of these spike EVs due to that EV decoy effect we mentioned earlier. So we standardize all of our input with our particle counts given to us by the MRPS technology. And also we used ratios, we calculated ratios of particles to add equal amounts of spike lentivirus and spike EVs in the conditions in which they were mixed. So to start, we see whenever we don't add any EVs, we get about a 50% neutralization. And when we add a 1-to-1 ratio of spike lentivirus and the spike EVs, we see a drop in the neutralization and a further drop in that neutralization when we increase the ratio of EVs to lentivirus to 2. Interestingly, we saw that our spike negative EVs increased neutralization levels for whatever reason, but the main takeaway is that we did see a decrease in the efficacy of this neutralizing antibody when we added in these spike EVs. So our takeaways from this experiment, our spike negative EVs unexpectedly increased the efficacy of a commercial neutralizing antibody. Our spike negative EVs also reduced infection by spike lentivirus in the presence of the commercial neutralizing antibody. And on the other hand, our spike containing EVs, they reduced the efficacy of the commercial neutralizing antibody and increased infection by the spike lentivirus. Of course, this was done with a commercial neutralizing antibody. And we asked ourselves, what about in a more physiologically relevant setting? We wanted to look at something with actual patient serum. So for this, we worked with a collaborator and obtained 9 convalescent COVID-19 patient serum samples. So patients who had recovered from COVID-19 donated their serum, and which should, of course, contain neutralizing antibodies against the virus. So we used these 9 patients. We made multiple dilutions of their patient serum, and then we repeated our assays, but we slightly changed things up a little bit. We used a different method of infection readout, where our viruses, if they establish an infection, they make the cells turn green. But everything else was done pretty much as before, where we mixed our lentivirus in with our EVs and our patient serum, and then we looked at the levels of infection. And again, our hypothesis was that, similar to what we saw in the commercial neutralizing antibody experiments, if we add spike-containing EVs, we will see a decrease in the serum efficacy and the neutralization and an increase in infection when we compare to the mock EV condition. So we of course did these experiments and plotted all this data, and I want to talk a little bit about something, how we calculated this EV decoy effect here, because this is important for all these plots that you can see. So the EV decoy effect here is calculated as the mean infection in the presence of spike EVs minus the mean infection in the presence of the spike negative EVs. Basically, if our EV decoy effect is greater than 0, this would mean that spike EVs led to an infection increase and a neutralization decrease. which is what we hypothesized. If our EV decoy effect is less than 0, then that would mean the opposite occurs. And if our EV decoy effect is 0, then that means the spike EVs had no differential effect. So when we actually plotted this EV decoy effect on the y-axis and we looked at the infection levels on the x-axis, we found that in nearly all of our patients, there was a positive EV decoy effect in the presence of spike EVs. This was pretty consistent across all of our patients. And this would kind of back up our hypothesis that the spike EVs led to a decrease in the efficacy of this patient's serum. And interestingly, there was a trend to these increases in the EV decoy effect. If the patient's serum by itself was lowly neutralizing or weakly neutralizing of the virus, the EV decoy effect would be even stronger. And if the serum was stronger at neutralizing to begin with, the EV decoy effect was not as pronounced. But nonetheless, regardless of how strong the serum was at neutralizing the virus to begin with, there was a clear influence of the spike-containing EVs on the ability of that serum to neutralize the virus. So our takeaways here, we found that SARS-CoV-2 spike-expressing cells can release EVs incorporating the spike protein. The majority of this EV-borne spike protein is the S2 subunit alone, but there is full-length protein present. Our spike-containing EVs increased the spike-mediated viral infection by reducing neutralizing activity of convalescent patient serum, and our spike EV effect is stronger with moderate to low amounts of neutralizing antibody and is presumably overwhelmed by higher amounts of neutralizing antibody in those high neutralizing patients. So just some future directions we were thinking about with this whole line of research. We wanted to know one of the things, which is what determines spike incorporation onto EVs? How is it determined whether an EV gets the spike protein? And if the EVs do incorporate the spike protein, how is the spike protein processed? How does it end up in just the S2 form versus the full-length protein? Additionally, we were curious what's the effect of the spike EV cargos and their effect on recipient cells. So in these EVs that are carrying spike protein, what kind of proteins or RNAs are they carrying, and do they have any special effects? And lastly, a very important question is, are these spike EVs detectable in the plasma of COVID-19 patients? Now, this isn't something we were able to ask ourselves in our timeline, and in my timeline at my former institution. But I did want to highlight that I did see an interesting paper come out recently that actually cited our article. And they did find indeed that COVID-19 patients did have circulating extracellular vesicles that carried the spike protein. And more interestingly, the levels of these extracellular vesicles carrying the spike protein were linked to the patient's outcome and how severe of an illness they had. So they basically found the more spike EVs you have, the worse you did against the virus, which kind of backs up what we were hypothesizing. Maybe this EV decoy effect is going on in these patients. So with that, I'd like to thank members of my former lab, of course, and any collaborators who helped out on this project. And again, I'd like to thank the webinar organizers for allowing me to give this talk. And I'll take questions at the end of our webinar. Thank you.
Thank you, Zach. That was a great presentation, super interesting. And I'll wait till the question period at the end. Dr. Sultana, the stage is yours. Thanks for joining us.
Okay. Do you see my presentation?
Yes.
Okay, wonderful. Hello, everyone. Good afternoon. First of all, I would like to thank Dr. Frekin and Lou Brown for organizing this Spectradyne webinar, and I'm very happy to have this instrument in our lab and using this routinely in our research. So I'm going to be talking to you about arthropod exosomes in vector-borne diseases. So we have done some of the discoveries or identifications, and we are pursuing more and more research in this area of arthropod exosomes, which is indeed a very novel line of research. So why do we really have to think and care about the arthropods or the vectors? So basically, the vectors or arthropods are medically important. So also we know that we ourselves, our children, our pets, our domestic animals, the wildlife, we all love to be outdoor, enjoying the nature's beauty and bounty, and hence becoming prone to the bites of those infectious arthropods, which do bring in the arthropod-borne diseases or transmit the arthropod-borne pathogens and bring in these diseases. They basically transmit these diseases to the humans and to the animals, thereby causing a big burden, a global burden all over the world. So when we look at the transmission cycle of a vector-borne pathogen, these pathogens basically persist for a very extended period of time in those arthropod vectors. They basically create a beneficial or a symbiotic relationship, which is indeed kind of hand-to-hand relationship where the pathogen survives in these vectors, the medically important vectors, for a very long period of time. And when these vectors bite us, they do transmit these pathogens, and we indeed, as accidental hosts, as humans, we do kind of see harmful diseases and the pathogenesis. So this is kind of not fair for us when in comparison to those medically important vectors or arthropods. So my lab has been working on some of the interesting vector-borne pathogens called flaviviruses. So these are basically viruses belonging to the family of Flaviviridae. And this genus includes the most popular West Nile virus, the dengue virus, which is a global pathogen, and the tick-borne encephalitis virus, TBEV, yellow fever virus, which we do have kind of some vaccine, which is kind of partially effective. And the recent outbreak with the Zika virus, I know we all forgot Zika virus because of the COVID, but it is still around there. And there are several other viruses that do cause encephalitis. And these viruses are basically named with the word flavus, which means yellow in Latin. And our flaviviruses are very small viruses. These are basically of nucleic acids of 10,000 to 11,000 base pairs. They have a positive sense single-stranded RNA genome. They are envelope viruses, and they are of sizes 40 to 65 nanometer in diameter. And we have to see them through a confocal or a electron microscope, such as a TEM micrograph here shown for the yellow fever virus. So most of these viruses are transmitted by the bite of an infected vector or the arthropod, such as a mosquito or tick, and hence they are also classified as arboviruses. So when we look at these vectors of medical importance, they are geographically being distributed all over kind of the United States. You see that the Ixodes cupillaris tick, which is also referred as the blackleg tick, is anywhere from the northeast to the south to the Texas. It's kind of expanding. And also the Aedes aegypti mosquito is also being kind of spreading all throughout. And you do see that some of the states like Virginia and Tennessee is being kind of been dominated by these medically important arthropods. So basically, ticks and mosquitoes are very important, and their transmission of flaviviruses is highly essential. So when we look at the diseases transmitted by these medically important vectors, such as ticks and mosquitoes, the ticks do transmit some of the diseases called Lyme disease, which most of you are familiar with. They do also transmit human anaplasmosis and other rickettsial diseases. My collaborator, Dr. Neelakanta, works on most of these rickettsial diseases, such as human anaplasmosis. My lab heavily concentrates or focuses on the tick-borne encephalitis, which is brought to us here in the United States by the Pauson virus, or in the Europe by the tick-borne encephalitis virus, which is the TBEV virus. We do also study some of the mosquito-borne diseases, such as the West Nile neuroinvasive disease, the dengue hemorrhagic fever, and the Zika-caused microcephaly, and the GBS, the Guillain-Barré syndrome. So we do kind of would like to address all these different diseases. So when we look at the vertebrate host, our research interest is basically lied on the mouse model. We do use a lot of mouse models in our diseases brought by the vector-borne pathogens. And we do also concentrate on working with several of the human samples, such as in vitro cell lines, primary cultures, as well as the stem cells and others. Now, being in the veterinary school of medicine, I am quite interested to expand my research to the higher level of vertebrate hosts, such as the sheep and perhaps maybe horses, because West Nile is very promoting in the horses. So we are very much interested. Now, some of the lab research interests from my laboratory are the novel pathogen transmission blocking strategies using the arthropod exosomes or determinants. And this is one of the funded study from NIH. We do also study neuronal exosomes. I probably may not have much time to talk about today about the neuronal exosomes, but there is a line of study that is being continued with neuronal exosomes that do facilitate West Nile virus and Zika transmission in the developing brains. We're talking about the mouse brains here. So we do work with some of the biomarkers because this is quite interesting, as some of our pathogens such as Zika and dengue do cause thrombocytopenia, which is a blood disorder with lower platelet numbers. And we do look for some of the biomarkers to kind of like diagnose this thrombocytopenia. We do work with some of the neural guidance factors such as semaphorenes, which are novel therapeutics perhaps in the flavivirus-induced neurological complications. My lab has been very motivated in looking for novel receptors for that facilitate the flavivirus entry in both arthropod as well as vertebrate hosts. We do kind of in and off work with some novel chemical complexes that could be used in flavivirus inhibition. And in overall, we would love to develop pan-flaviviral vaccines that would be using either viral factors as well as the host factors. Now, in collaboration with Dr. Neelakanta, I do study rickettsial diseases, which is another funded study from NIH. And maybe another time, Dr. Neelakanta may present that part. I'm not going to be working on that now. So, but basically, I would like to briefly talk to you about the arthropod exosomes and maybe a little bit on the neural exosomes. So as I said, this is our funded study. We have been working on this quite extensively. I probably may not have to give a background for the exosomes or extracellular vesicles, assuming that the audience here are mostly with all this background, but just briefly letting you know that exosomes or EVs are small membranous bound microvesicles. These could be anywhere from 30 to 150 nanometer, referred as exosomes, or if they are higher in sizes, going up to 400 to 500 nanometer, they could be referred as extracellular vesicles or EVs. They are mostly of endocytic origin. They are formed in the late endosomal compartments, and they do contain a lot of stuff in them. That's why they're referred as signaling related vesicles. They do contain the nucleic acids such as DNA, RNA, and microRNAs, several proteins, lipids, and whatnot, cell surface receptors, and stuff like that. So what you see here is the endocytic pathway that is being kind of taken up of material by the transferrin receptors, leading to the formation of early endosomes and late endosomes and the intraluminal vesicles that would again lead into the formation and release of exosomes, which are basically small membrane-bound vesicles of 30 to 100 nanometer in size. Now, this is what an exosome would look like. It is being decorated with lipid rafts on the surface. There are several cell-specific receptors and tetraspanning domain containing proteins such as CD63 and CD9. And indeed, it has also a lot of luminal content that is being packaged into these small vesicles called EVs. So we did hypothesize, does EVs or exosomes facilitate the flavivirus transmission from these medically important vectors such as ticks and mosquitoes? It was a big question in those days when we asked this question in 2014 and 2015. And we did indeed work a lot, struggled a lot to publish our work, but finally, indeed, we did made our way to get through this. So we also asked the question, are exosomes important means of transmission from the vector host to the vertebrate host? Because remember, we're working on vector-borne diseases that do get transmitted from the infected vector to the vertebrate host. And we do believe that these could be envisioned as transmission blocking strategies because the flavivirus would exit the arthropods and infect the vertebrate host, perhaps using these EVs or exosomes. And we did kind of raise this question and envision this as one of the best approach to develop the transmission blocking vaccines. And I'll come to what are transmission blocking vaccines. So we basically concentrated under the umbrella of host pathogen interactions, where we looked at the modes of transmission and the pathogen shedding. So we, of course, use multiple approaches to answer these questions under this umbrella of host pathogen interactions. We use the molecular biology, genetics, biochemistry, cell biology, virology, neurobiology. Of course, we did a lot of bioinformatics to kind of simulate our analysis. And then we also use mouse as the model to understand the pathogenesis. Now, I'm not going to go over all the published literature, but I would just like to tell you some of the breakthroughs from our research. So we did publish a work in PLOS Pathogen that was featured as a featured article that was highlighted in several places. And this is all work about the tick-borne flavivirus transmission called Langat virus, which happens through the tick exosomes or the arthropod exosomes. And we did indeed also presented another work where we looked at the mosquito-borne flavivirus dengue transmission through the mosquito exosomes. And this work has been published in PNAS. And these are both articles from 2018. So basically what we overall did is we look for the isolation of EVs or exosomes from either the tick cells or the mosquito cells. And shown here are the cryo-EM images, which has been performed at our collaborators facility at the UTMB, Texas, Galveston, Texas. So these are exosomes from uninfected tick cells or LGT infected tick cells. You see that these exosomes are all of heterogeneous in population. They are all of different sizes and diameters. We did indeed run also the density gradient prep, where we isolated 6 different fractions from our EVs or exosomes, either from uninfected tick cells or LGT infected tick cells. When we probe these fractions for our viral envelope protein, you see that our viral envelope protein is abundantly being enriched in this fraction 4 and 5. And indeed, it was present in all different fractions. We do see kind of some faint band in fraction 1 and 6. However, it was kind of very enriched in fraction 4 and 5, suggesting that perhaps the viral cargo or material is being selectively being sorted or transported in those vesicles, which could be of very small diameter in size. We assume that because these are the fractions collected from top to bottom, and these EVs or exosomes could be of sizes anywhere from 50 to 100 nanometer in size. So when we looked at the loads for our other viral proteins, such as from dengue serotype 2 or dengue serotype 3, you see that we do detect the presence of viral envelope protein in exosomes. However, you do also see the presence in the cell lysates. We do also see the presence of NS1, which is the non-structural protein 1, and dengue serotype 3 perhaps indeed mimic the dengue serotype 2, where we could see that both these viruses have their envelope proteins being contained in the EV cargo as the EV cargo. We also were able to show that the Zika envelope protein is presented or enriched in fractions 4 and 5, and this was also being coinciding with the heat shock protein 70, which is also kind of been abundantly been packaged in the fraction 4 and 5, suggesting that these are the fractions basically could be referred as exosomes. So indeed, now I'm trying to tell you that we have envelope proteins from our viruses, flaviviruses like Zika, dengue, or Longut virus. Now, what happens to the genome? This was very interesting study where we could see that the mosquito-infected cells that derive the exosomes or EVs do have full-length dengue 2 RNA genomes contained in those mosquito exosomes. So we basically designed the primers for the entire genome. We amplified the products, and this is the amplification done from the mosquito cell-derived exosomes or EVs. So you do see that we are able to amplify the entire dengue 2 viral RNA genome. So this brings a kind of a big step in or breakthrough into the community, because indeed the infectious EVs are really infectious. We don't need the viral particles or the virions or the naked virus particles. So the infectious EVs are indeed enough to bring in the infection because they do contain the full-length RNA genome. And indeed, this was true for several of our viruses. I'm not showing you the data, but we do have been successful in amplifying the RNA genomes from Zika, dengue, West Nile virus, and different of our flaviviruses. So basically, this is a relationship, something like a virus being sticking with the exosome so that it can be nicely transported. So currently, we are addressing the specific packaging and transport mechanisms and the cargo going in the tick and mosquito-derived exosomes. So there is a lot of genomic and proteomic work being done in our laboratories to understand the cargo and the packaging mechanisms. So when we look at the tick cell-derived EVs here, of course, they're heterogeneous, as I showed you, and they're mostly of sizes 50 to 100 nanometers. So this is basically the EV quantification from uninfected tick cells or LGT-infected tick cells. So when you look at the production and release of EVs, you do see that in presence of infection, we do have higher production and release of EVs. And then we ask a question, is there anywhere sphingomyelinases from ticks are involved in this higher production and release of vector EVs or the tick EVs? We did publish another study in Frontiers in Cell Microbe, where we were able to kind of pull out one of the ortholog of a poisonous ortholog from the spider, a venomous spider, Loxilus, where we were able to find that this molecule plays indeed a very interesting antiviral role. So I'm not going over the data from that study, but I'm just proposing the model, what we showed. So what is basically being shown is the Longut virus, which is a tick-borne virus, replicates using the sphingomyelin lipid, which is basically accumulated in the peripheral membrane or in the EV membrane. And this virus basically inhibits the ISS-MACE, the venomous spider ortholog. And basically this happens by the accumulation of the sphingomyelin lipid. So this free sphingomyelin leads to the production and release of the EVs that do contain the viral content perhaps. And when we treat the cells with GW4869, which is a pharmacological agent that do interfere with the production and release of EVs, this inhibits the viral loads and the sphingomyelin accumulation. And it indeed also restored the ISS-MACE levels, activity, and it also inhibited the sphingomyelin support in the EV formation. So basically our molecule ISS-MACE acts like a hero. It's like an antiviral molecule that do kind of inhibit the viral replication and also the viral association with the sphingomyelin lipid and the sphingomyelin lipid participation in the additional or the enhanced production and release of EVs intakes. Now I'm moving forward to another study that perhaps is also one of the breakthrough study. I'm going to talk to you about the role of in vivo exosomes from the tick saliva and salivary glands. And this is indeed a very challenging study because getting saliva sample from ticks is indeed very challenging. You and I may salivate as we smell nice food, but ticks are very, very clever and they wouldn't be salivating easily. We have to like partially feed them. And even if we collect some saliva, it would be less than 10 microliters. You can imagine we may have to really work hard to get this out. So when ticks feed, this is the feeding cavity here. So you see a pit here, right? The tick is almost kind of partially being immersed into this cavity. Now, what is happening in this cavity here? There's a lot of things happening. There is a skin injury, there is wound healing, there is also like inflammation, pain, and there is accumulation of the immune response. And of course, indeed, there is also transmission of pathogen. And we do believe that when ticks salivate and the saliva indeed bring in those infectious exosomes that indeed would bring in the transmission of pathogens to the human skin or the vertebrate skin. And indeed that may get into the blood capillaries or the lymph nodes and bring in the infection to the vertebrate host. And this is also been kind of like being shown as a proof of principle in our study, PILOS pathogens, which is published in 2018. Now, in this study, we did also image the in vivo exosomes that were derived from the ambulum mammaculatum tick saliva or the salivary glands. We are using two different ticks here. Another tick is the Exodus cubellaris ticks here. I'm showing you the cryo-EM images from saliva or salivary glands that were basically performed at two different institutions, the University of Texas Medical Branch, the UTMB, or the Indiana University. You see that again, exosomes are all, or EVs are all heterogeneous in population. We do detect the presence of the CD63 and the heat shock protein 70 as the EV markers from the saliva or the salivary gland derived exosomes. And these are 6 different fractions from the density gradient. And we do also have salivary gland derived exosomes from the Exodus cubellaris ticks, which are the popular ticks for the transmission of the Lyme disease and other important diseases like rickettsial diseases. And we do also have in control as our ISE6 cells. These are the Exodus cubellaris embryonic tick cells that do also show the presence of the EV markers. We are now characterizing some of these additional bands that we show here as our proteomic analysis. So what comes here next is the interesting part of the data for those of you who are interested in the Spectradyne device. So we did use the Spectradyne nanoparticle analyzer. And I'm showing you some additional cryo-EM micrographs here, which I'm not going to go in. So basically, I'm going to discuss the data shown in B and C. So what I'm showing you here is the particle diameter measurement analysis against the concentration of particles to determine the exosome sizes from either the ambulum mammaculatum saliva shown in B or the Exodus cubellaris derived exosome shown in C. And basically, the N represents the particles determined or counted using the filter TS300 that was purchased from Spectradyne, and this basically measures EVs or exosomes of sizes 50 to 300 nanometer in diameter. And the scatter plots basically on right in both B and C shows the number of particles counted during the transit time. So this is what is being shown here, and this is indicated as N. So we have basically used the Spectradyne NCS1 nanoparticle analyzer. We currently have this device in our laboratory. Both Dr. Neelakanta and myself use this. This is not just for the EV folks, but this can also do a lot of terrific amount of work. We do count the number of bacteria in our growing cultures or supernatants. We do count the number of platelets in our cultures. It does a lot of fascinating stuff. So I do strongly recommend the Spectradyne NCS1 nanoparticle analyzer. Now moving on, what we did next is we took this tick cell or tick saliva or salivary gland derived exosomes, and we did perform some scratch-based assays, the wound healing assays, and we were able to show that when we keep the human skin keratinocytes, the Hackett cells, so this is the group with untreated, no exosomes were added here. However, the groups here were added either with the saliva-derived exosomes or the salivary gland-derived exosomes. This is the salivary gland-derived exosomes from another tick, Ixodes cupillaris, or the cell-derived exosomes. So when we add these exosomes to our human keratinocytes, the Hackett cells, you see that these cells could not close the wound. They could not close the gap between the two different side of the monolayer. However, the uninfected, within 24 hours, it could close the gap. So the wound was not able to close. So this suggested that the tick exosomes basically delays the wound healing process and the repair process in the human skin keratinocytes. And this is basically the quantification of what I show you here with the images. So you do see that the untreated group closes the wounds. However, the tick exosome derived groups do not closes the wounds. Now, what is basically happening at the skin barrier when ticks are feeding? Basically, there is a lot of immune response, as I said. There is amount of cytokine response. So we next went ahead and looked at the multiple cytokines by purchasing some cytokine arrays. You could see that they were different. Some of the cytokines were differentially regulated. And we were able to find that one of the interesting molecule was CXL12 and IL-8 that was significantly being modulated upon tick exosome treatment on the human skin keratinocytes, not just by the wound healing assay, but we also could find that this was indeed true by the cytokine array as well as the quantitative PCR analysis. So what I'm showing you here is that when we just add 2 microgram of CXL12, I'm pressing this again, just 2 microgram of purified protein, CXL12, in addition to those exosome treatments, we were able to now find that there is a closure of the wounds, or there is a repair process that is happening, which is quite comparable to the untreated group. So you see that within 24 hours, the untreated group closes the wound. You see some kind of gap here still in 16 hours. However, the gap is being closed by 24 hours. Now, when you look at the group with the CXL12 and also exosome treatment, you see that all the groups did close the wound, suggesting that CXL12 is very important molecule during the tick feeding, which would kind of help in the skin injury process and the repair process. So we did a lot of work on that. I'm not going to go over. This is also another very interesting published study. I'm summarizing this work here. So what we believe is when ticks feed, they do salivate and secrete the saliva. Now, saliva brings in the exosomes. The exosomes kind of downregulates the CXL12 and upregulates the IL-8. We believe that the downregulation of CXL12 basically leads to the less local cell proliferation or migration or inhibition of angiogenesis, and this leads to the delayed wound healing. But however, with purified CXL12, it corrects or restores the process. The entire process is being restored. The IL-8 induction upon the tick exosome treatment could increase the wound healing. It could be the effect brought from the tick side, where tick exosomes are inducing the IL-8 or the CXL8. So this is an interesting study, and we have been pursuing a lot more to understand what in the tick exosomes is perhaps doing all this. We are trying to understand the mechanistic inside, the further details. So please catch up with our literature. So some of the novel findings from our studies is our lab is the first to show and study arthropod exosomes. These are exosomes basically from ticks and mosquitoes, or the mosquito and tick cell derived exosomes. We have been able to show both positive and negative strands of the viruses, the flaviviruses, and the full-length RNA genomes being contained in these arthropod exosomes. We do also have seen the presence of viral proteins such as the envelope and the NS1 proteins. Indeed, we have also shown the presence of the complete polyprotein being contained into these infectious exosomes. They have been kind of very nicely been characterized. We have used several of the viable assays where we have used like clot formation assays, the reinfection kinetics, and transmigration assays. We have been able to show that the inhibition of exosome production basically hampers our viral transmission and the viral loads. We have also indeed identified a novel arthropod exosomal enriched molecule called TSP29FB, which is a tetraspanning domain containing protein and ortholog of the human CD63, which indeed shows a lot of effect. Please catch up with our literature on PNAs. And we do believe that either inhibiting TSP29FB or inhibiting exosome production and release could be the novel modes of therapeutics for blocking the transmission of vector-borne diseases. We do also believe that the tick exosomes do modulate the cytokine response and wound healing process. And this could be used of, and the CXL12 IL-8 could be used as therapeutic molecules at the tick-human skin interface. Now, this is the summary of all our work overall. So when ticks do feed or mosquitoes do feed, they do contain the infectious exosomes that are secreted through their saliva. Now, these exosomes do come in contact with either the skin barrier or the endothelial barrier, and they do infect these two barrier cells, leading the infection to the peripheral tissues such as spleen. Now, high infection or viral loads of viremia in the spleen and other peripheral tissues such as liver would breach the blood-brain barrier, leading the infection to the brain, causing neuroinvasion and neuropathogenesis. And of course, this leads to encephalitis and ultimately to the death of the vertebrate host. So this is basically the summary of all the work being presented today. Now, just briefly, just touching base on the neuronal exosomes, we have done some beautiful work with Zika virus, where we were able to show that the Zika virus induces the cortical neural loss in the time and dose dependent manner. So you see the time course here. So as we keep the infection ongoing for 120 hours, basically 5 days post-infection, you see that the neurons are dying compared to the uninfected control group. This is basically quantitated by the MTTSA. So here I'm basically showing you that Zika virus infected cortical neurons were highly susceptible to infection. As you see, when we stain for the MAP2, the microtubule associated protein 2, which is a marker for the neural extensions or dendrites or axonal structures, you see that they are basically getting diminished at the site of infection. The infection is shown in red, MAP2 staining in green, and you see in the merge that the neurons that are infected basically have no staining for MAP2, suggesting that the neuronal extensions are basically getting diminished. So the overall summary of this work is suggested that Zika virus basically induces the neutral sphingomyelinase 2, the SMPD3 molecule, which basically depends on a sphingomyelin for its function. The sphingomyelin lipid is converted into ceramide and phosphocholine by SMPD3. And this basically leads to the production of exosomes or the release of exosomes. When we bring in the GW4869, it basically inhibits the SMPD3 and the production and release of exosomes. And indeed, if more exosomes are produced, which indeed is seen in neuronal exosomes with Zika infection also induces the production and release of exosomes. So this again facilitates the Zika infection and the dissemination of infection throughout the brain, causing total neuronal loss and death. So where all our study is leading to, basically we believe that our study is going into the anti-vector vaccine studies, where we do believe that we can generate vaccines against the tick and mosquito-borne pathogens. How does this work? So when we administer the transmission blocking vaccines into humans, so these vaccines are basically being produced they bring in the antibody response in the circulation, and when they are circulating in our circulatory system and uptaken by these medically important arthropods or vectors such as ticks and mosquitoes, they take up these antibodies into their blood meal. And what happens when they take up these into the blood meal? So basically these antibodies would block the parasite transmission from the midgut of these arthropods to the salivary glands. So for the transmission of the pathogen within the vector, all the pathogens have to be brought from the midgut to their salivary gland so that it's secreted into the saliva. Now, if we can block this parasite transmission or the pathogen transmission from the midgut to the salivary glands, that would be a bingo. We would be stopping the pathogen transmission from the vector to the human side. So this is quite challenging, and this has been quite outspoken in the field, but not really serious efforts have been done. My lab and Dr. Nilakanta's lab have been seriously been concentrating on this transmission blocking vaccine strategies. And with that, we can invite the pathogens to take a fight with us. Perhaps we would be winning against all the different pathogens. So our research is basically like a fight against the arthropod-borne viruses. We have been trying to use approaches from virology, medical entomology, and vaccinology to bring in prevention, treatment, and eradication of these vector-borne diseases. So these are my current laboratory members. I have 3 dedicated postdocs and graduate students and an undergrad. They all are hardworking researchers. And I have trained a couple of people from my lab, some of the postdocs as a co-advisor and a postdoc as a major advisor. And the work I presented is mostly from my very motivated PhD graduate students. The PNS work is from my first graduate student, Ashish Ora and Venshizu, who did a really terrific amount of work. I had no time to present the work on thrombocytopenia from my other graduate student, and some of the undergraduates have also done terrific amount of work. And we had an army of undergraduates back at Old Dominion University in Virginia, and we think that they are a very important part of the lab. So I would like to also acknowledge my long-term collaborators, Dr. Neelakanta, and some of my advisors, Dr. Vaikal, Anderson, and Wunderlo, and several of the collaborators, especially Dr. Fatah Kashanchi, who is a pioneer in the EVs field. I would definitely suggest you guys to look on his literature. He's been really a guru in the field of EVs and has been very interesting colleague. I would like to also acknowledge the funding from NIH and the funding given to me from the University of Tennessee at Knoxville, a startup, and also the ODU startup funds given to me, which helped me lead this work. I would like to always acknowledge these three important institutions in my career: the Yale for giving me or seeding the training in me, the Old Dominion University for giving me research independence, and UVA for extending my research back at ODU, and now the University of Tennessee for further enhancing my research program here at UT. Thank you. With that, I would be happy to take any questions.
Thank you, Dr. Sultana. That was a really interesting presentation. There's so much to unpack, and I think really highlights how much there is to learn about EVs in general, you know, even in just a small niche like the arthropod vectors. That's really cool. I would like to just before we get into questions, summarize that. I think I need you to stop sharing. Yep. Okay, perfect. Thank you. All right. So I think both our presentations today really highlight a lot of great examples where EV concentration was important to the research, right? And it enabled careful analysis of the data and stronger outcomes from the experiments. Thank you for the extended plug, Dr. Sultana. Our technology does this, right? And if you would like to learn more, please come to our website about it. I will hand over the Q&A session very quickly to Inal, who has more skills than I do for managing questions. Thank you very much to both of our speakers and to all the attendees that came.
Thank you very much to everybody. Thank you to Spectradyne for sponsoring, and to Zach and Dr. Sultana for fantastic presentations. Ladies and gentlemen, if you'd like to ask questions, the best way is to raise your hand electronically. And we already have somebody who raised their hand, and we'll unmute you so you can ask your question in real time. So Tatiana, now you can unmute and ask your question. Welcome.
Hello, can you hear me?
Yes, we can.
Sorry, I just had to change my microphone. I'm not sure if it's still working. Is it?
Yep, it's working. We can hear you. Yep. Go ahead.
So I have a question for Zach. I got the impression that you did not really do validation methods to say that the excellent.
Yeah, saying it's a hypothesis. So do you have any plans to do any validation methods for that, like to visualize the interaction between the exosomes and the antibodies or not?
I think your question cut out a midway through, but I think you were asking if we had plans to look at the interactions with the antibodies with the extracellular vesicles. Was that your question?
Yeah.
So I'll not currently, because this was work done during my PhD, and I've kind of taken on some new projects in my postdoctoral fellowship. So at the moment, we're not planning to, but of course, that would be something very interesting to actually visualize the interaction somehow. I think that could be pretty easily done if we did some sort of immunogold EM staining with the EVs and the neutralizing antibodies. But no, we didn't do that in this paper, and I'm not sure if that's going to be something that's done in my former lab. But that's all I can say for that.
Okay, thank you. It's because I'm doing nanotherapy with exosomes and cancer cells. So I was wondering if you can from your experience, say what kind of methods you would think of, but you just mentioned them. So yeah, that was the question.
Yeah, if you're thinking of a visual interaction, I think immunogold EM would be at least something that would show that the antibodies are actually binding to the EVs.
Okay, thank you.
Great, thank you. So there's a question that came in via the Q&A box, and I'll read it out. And this is again for Zach. And it is, how long after recovery was the serum drawn from the COVID-19 recovered patients? Was there a correlation between the draw time point and infection response? Are there plans to get more patient samples to study this potential correlation? So Zach, would you like to take that?
Sure. So the convalescent serum was taken anywhere from 45 to 110 days after symptomatic infection. We worked with a collaborator to get this serum, so that's the information that they gave us. I didn't have individual data for each patient upon with how long after their infection they were drawn, and therefore I can't say for sure if there was a trend in the ability of that serum to neutralize and how soon after the infection it was taken. Although I would suspect that those serum samples that were taken closer to the symptomatic infection are likely the ones that were higher neutralizing, but it's hard to say for sure because I don't necessarily have that info, and I'm not sure if there are plans to recruit more samples, because again, this was a collaborator who was getting us these samples, so it was their IRB that was getting these patients. I think they were planning to publish some sort of paper using a lot more serum, so but I can't really give too much info about that. I can just say that the PI's name was Christopher King, and he's at Case Western Reserve University.
Thank you very much. There's two questions in the chat box that came from Dr. Chu. So Dr. Chu, you can now unmute if you'd like to ask the questions in real time, and you're welcome to ask them. I can also read the questions out if you prefer that. And the first question is, does CS1 measure particles bigger than 10 microns? And then perhaps Jean-Luc, you want to take that one?
Sure, yep. Currently our upper size limit is 10 microns, so the answer is no. But there are a lot of technologies out there to measure larger particles too.
And the follow-up question from the same participant is, does CS1 differentiate different types of particles, for example, protein particulates versus other types of particulates?
Good question. So the answer is the NCS1 currently measures just the size and the concentration of particles, so it just counts them, but stay tuned. Okay,
thank you. Ladies and gentlemen, we still have time for maybe one or two more questions. We're right on schedule. So if you'd like to ask the question, this is a great time to ask this panel of participants, both from industry and academia. Any questions you might have, you can raise your hand. We'll unmute you and you can ask them just like a real conference. Okay. Well, I think at this point, we don't have any further questions. We would like to thank the Spectradyne team, Dr. Jean-Luc Frekin and Lou Brown, for bringing together this webinar. We're delighted that they participated and brought it to fruition. And we look forward to their participation at many conferences. I know Jean-Luc will be speaking at several events and always engaging to hear the technology platform. Also, this will be available, this recording, as a video on demand. So please watch the Spectradyne website. I'm sure they'll do an emailer when the video is uploaded. So you can look at some of the slides, which will be submitted by Zach and Dr. Sultana. So we'd like to thank everybody, thank our guest speakers, and we'd like to close this webinar at this point, and I hope everybody has a great day. Thank you very much.
Thank you very much.
Thank you. Goodbye.